The immune system is perhaps the most difficult system to teach because there are so many specific connections to be made between fairly complicated stories. So it's always those connections that are the most difficult to make, especially for information that you've just learned. All right, so we're going to start off with a pathogen. The typical definition for a pathogen is that it's a harmful or disease-causing organism. We typically think of a bacterium or a virus. But in fact, a pathogen is anything that is non-self. Think about transplanted tissue or a transplanted organ into the body. Our immune system tends to reject it. So if it's non-self, that means it doesn't belong to you. And what actually defines something as non-self are these antigens, these proteins that usually stick out from the surface of the membrane. These little proteins here, we're going to call them pathogenic antigens. Sometimes they're called non-self antigens. And that's what makes something an actual pathogen. The body recognizes it as an enemy and begins to attack it. So I'm going to be using the color red to describe all of the pathogens today. And we're going to describe 3 different levels of defenses. In green, we're going to do the first line of defense, or the innate immune system, outside external structures. The second line of defense is going to be in blue, and the third line of defense is going to be in purple. So we're going to start off, of course, with our external defenses. Our first line of defense, these are just things that are just in place that pretty much keep all of our pathogens out. We start off with skin. Those keratinocytes in the epidermis of the skin do a really good job of keeping stuff out. The acidity on the surface of the skin, along with some other substances such as dermcidin or bactericides that are part of your sebaceous or sudoriferous secretion. So there's a lot of things on the surface of the body that kind of tamp down the number of living organisms living on your skin. My sad little representation of a face there. I did want to point out lysozyme. Lysozyme is secreted by the tears and in the saliva, so eyes and mouth. Lysozyme, as the name suggests, is an enzyme that lyses things. To lyse something, L-Y-S-E, is to break it apart. So lysozyme is something that is present in these 2 places, kind of special, that pretty much nothing can live there. It'll pretty much just lyse anything that it comes into contact with. It breaks apart everything. Right, so now we draw, in the anteriorly, we're going to draw our windpipe, the trachea, and posteriorly, our tiny little esophagus that's kind of collapsed and soft. And these are going to be representing 2 structures, which we're going to be exploring soon: the respiratory system and the digestive system. Both the respiratory system and the digestive system are lined with mucous membranes called mucosa. We actually studied these in the previous lecture when we talked about lymphatic follicles, lymphoid follicles that are buried in the mucosa. But of course, as the name suggests, there's also mucus secreted by the mucosa. So the mucosa will line these tubes, such as the esophagus, and then the rest of the digestive tract, like the stomach, the small intestines, large intestines. There's a mucosa extending down the whole thing. And then the same is true of the respiratory system. You'll have the trachea lined with a mucosa, but also the bronchi, primary, secondary, tertiary bronchi, and even the bronchial ducts, or the ducts going directly to the alveoli themselves. So all of these structures have a mucous membrane, and so they secrete mucus, and the mucus helps to trap pathogens that come along through the food that we eat, digestive system, and through the air that we breathe in, respiratory system. The mucus will trap these pathogens, and then cilia are oftentimes present that will kind of brush them along, especially in the respiratory tract. It kind of brushes them along and moves your mucus back upward to ensure that you are getting rid of those substances. So the mucous membranes are highly effective. Additionally, I just wanted to mention, kind of famously, especially the stomach in the digestive tract is going to be chock full of acidity. So we mentioned the acidity on the surface of the skin, the acidity which is really intense inside of the lumen of the stomach, but also there will be some acidity, for example, in the vaginal tract. And we'll talk about that more down the line. But the idea about acidity is that most things can't live in an acidic environment. So a pH of about 6, like the surface of the skin has, or a pH of 2.5, which the stomach has, it would be very difficult for most living things to exist in that environment. So that acidity itself, it helps to kill off most everything. Right, so now we're going to go back to the idea of the surface of the skin because this is a good place to talk about kind of the penetration. So a lot of ways that that pathogens will attack your body is through the skin, such as what we're drawing here, like a little thumbtack that you accidentally stick through your skin, for example, or through the digestive tract or the respiratory tract. These are the typical places where you get sick. So what we're drawing here is probably the simplest way to picture, to visualize how that actually happens. You will literally have bacteria that were on the surface of that thumbtack penetrating into your body, and they start to basically float through your interstitial fluid. Additionally, some of those pathogens that were on the surface of your skin will also come along and kind of penetrate your body that way as well. Let's switch to the blue marker here because we're going to be starting to take a look at our second line of defense. Our second line of defense is going to be what is in place when that first line of defense is breached. So briefly, we're going to simply describe fluids, chemicals, the inflammatory response coming along and making a difference. And we'll kind of talk a little bit more about how that works in a moment. But I will say also that all these extra fluids are going to be drained by the lymphatic system as we described in the last lecture. So the idea is trapping pathogens in one area and then draining them through the lymphatic system where you have lymph nodes and things like that set up to monitor for issues. Additionally, what we're drawing here, kind of hidden by my hand, are phagocytes. And the phagocytes we're specifically drawing are, you may recall, neutrophils, the most numerous of the white blood cells. They are granulocytes. And so as you can kind of see here, these neutrophils begin to actually consume the substance. So the first two things that happens when you are penetrated are you'll have phagocytes coming along and start to consume the actual pathogens, whatever they are, and you'll have that inflammatory response, which we'll discuss in a moment. So let's take a look at phagocytosis. Phagocytes would be, for example, macrophages, dendritic cells, and neutrophils. There are other phagocytes as well that are actually capable of consuming things, but we don't dwell on them too much. So these are the three big players. The root phago means to eat, or I think it actually literally translates as stomach. So a phagocyte is a cell that consumes things. So let's take a look at how that works. We're going to have some pathogens, whatever this pathogen happens to be. Let's say that it's a virus, comes along, and the phagocyte goes after it, the neutrophil goes after it and begins endocytosing it, bringing it into its own cytoplasm, but it's not going to be affected by this because it's going to be surrounding it by a bubble, by a vesicle that protects the actual phagocyte, the actual neutrophil, from the damaging effects of this virus. So off to the side here, we'll draw a little lysosome. Lysosome, S-O-M-E means body, and L-Y-S, again, means to lyse or to break something apart. So this is a little body, a little organelle inside of the cell that breaks things apart. It uses acids and enzymes to do it. It's basically a little stomach inside of the cell. Now, it turns out that bulbul that is surrounding the virus is called a phagosome. So it's an eaten body. So it's an endocytosed body that includes that pathogen. So what we're going to do then is we're going to bring together these two things. We're going to create a phagolysosome where the acids and enzymes inside of the lysosome are going to combine together with that pathogen and break it apart. And then it's released harmlessly. There's actually one other thing that can be done here, which we'll be discussing in a little bit. But this is a pretty simple way of describing the story that we'll just kind of stop it there. And we'll come back to this story in a little bit. But the idea is that the contents of the lysosome combine together with the contents of the phagosome, acids and enzymes combine together with that virus, pathogen, to create a phagolysosome, and this in turn breaks it apart, and then all these particles are released harmlessly into the interstitial fluid, and then eventually released from the body. All right, let's go back to our story of inflammation. And there's a lot to say about inflammation, but we'll describe it as simply as possible. Inflammation is redness, heat, and swelling. This is enhanced by chemicals called cytokines, which you notice we kind of wrote chemicals up above there too. So the main purpose of cytokines, at least for our purposes in this lecture, is to attract activated T lymphocytes and activated macrophages. And once these cells are activated, these are white blood cells that have a number of purposes, but one of the things they're able to do is to release more cytokines, which in turn enhances the production of T cells and macrophages. And so this is a positive feedback cycle. The more cytokines you have, the more of these white blood cells you have, and other white blood cells as well, but these are the big players. And the more of these white blood cells you have, the more cytokines you have, and so on and so forth. And I will also say just the practical aspect of this is that the way that you have redness, heat, and swelling is the vasodilation of arterioles and making those capillaries leaky. We've mentioned this a few times now, but I just wanted to make sure that we're kind of combining all of those stories together. So much more to say about inflammation. It's painful not to say more, but that is what we're going to say for now. All right, so just to recap, our first line of defense was our innate, or kind of our external defenses. And our second line of defense, two aspects of which we've discussed so far, would be internal and nonspecific. These guys do not care what the pathogen is. All they know is that it is non-self, and therefore they're going to go after it. The inflammatory response will begin, the cytokines will come in, all those fluids and all the blood vessel stuff will happen. And also, on the other side of it, you will have phagocytes immediately coming in. Now, all these things will happen literally within seconds. You can have a good inflammatory response that begins within about 30 seconds. Those neutrophils are pretty much always present, so they are one of the big players that begins phagocytosis immediately. So these are the first two big players in the second line of defense. We're going to go ahead and label and just briefly talk about three other aspects of the second line of defense. Natural killer cells. I've actually been seeing more and more information about those natural killer cells. NK cells work to perforate anything non-self. They basically put hole punches in the membrane and break it down. Fever. A fever helps to speed up your metabolism, which helps to keep your body's cells going faster and healing faster. It also helps to sequester zinc and iron in the spleen and in the liver, kind of hide them away. So you feel a little bit run down when you're doing this, but you also help to take these necessary nutrients away from any kind of microorganism that might also want to utilize them. And then there's complement. Complement is 20 or more proteins, 30 more proteins, somewhere, a large number of proteins that actually stack together and lyse or break apart any kind of a non-self structure. They also work through opsonization, which is where they attach carbohydrates to the outside of the structure, and it's literally a sugar coating. And this actually helps certain things to be more readily recognizable, so that, for example, phagocytes would be able to come into the area and more easily recognize the enemy because of that sugar coating and all those signals there, and that would help them to do their job better. So, yes, so the second line of defense so far, we've talked about phagocytosis, inflammation, natural killer cells, fever, and complement. And then the last thing we're going to be discussing today is in terms of the second line of defense are interferons. So interferons, we're going to do a little sketch for these because it's a little bit more involved. Interferons are specific to viral protection because of the way that viruses actually work. Viruses are not actually living things. They cannot replicate on their own, so they require a host to do this. So welcome to the world of viruses. The reason they infect us so easily is because they need us to survive. So we're going to draw two cells of the lungs. These are simple, some kind of epithelial cells in the lungs. And what we're going to do is we're going to draw just a really simple little sketch of how viruses actually work. They attach themselves to the outside of the cells usually, and then they inject their little DNA or RNA, and then they manipulate the host cell into making more of them. And all of a sudden you've got like 200 little viruses running around inside of the cell. So although I'm not drawing this, have an understanding that there's a nucleus in there, and you've got, your RNA or RNA polymerase, and you've got all the ribosomes, everything involved in actually creating new proteins. That's what's being used by the virus to replicate more of itself. So it turns out a virus is a really simple structure. It's not a living thing. It cannot replicate itself. So this is one of the definitions of life, is that you'd actually be able to replicate. Viruses cannot do that. They are basically a coating of proteins, and inside of the proteins would be this DNA or RNA, which in turn, that's pretty much all they need. They just inject their little nucleic acid, their little DNA or RNA, into a host cell, and this is how they make more of themselves. Well, so this first cell here that has been infected is a goner. Most cells will not survive this type of viral infection. There's just too many viruses. They're going to take over. They're going to burst the cell from the inside. But the original cell can save its friends, basically, by releasing interferons. So interferons is our last topic that we're discussing of the second line of defense. Interferons, it doesn't matter what the virus is, it's still nonspecific, but if there is a virus there, be it Ebola or influenza or coronavirus, anything like that, then it will, the infected cell will go tell its friends by releasing these chemicals called interferons and warn them away. And basically what they do is they say, stop producing proteins for a little bit. Stop any kind of replication of proteins, stop all of your cellular machinery, just stop producing proteins, because if these viruses come along, and here they come, and they try to manipulate you, maybe you have a chance of surviving. So one of the reasons why viral infections make you so run down is because these interferons actually will kind of shut down your cells, but it's for a good reason. You can see that those viruses that tried to infect this cell were not successful because the cell had shut down its own protein producing mechanisms. These were unable to infect that cell. So that is the purpose of the interferons, specific to viruses because of the way that the viruses manipulate the host cells. Right, let's move on to our third line of defense. Here we're looking at internal and specific. We're going after a specific pathogen in this case. So let's take a look at how this works. And this is where we interact the second line of defense with the third line of defense. What we're going to produce here is the phagocyte is going to, remember that we said earlier that a phagocyte is capable of simply releasing structures harmlessly, but another thing that it could do is it could actually present the pathogen, as we see here, that little bit of red, just a little bit of the membrane of that pathogen. And when this happens, it becomes an APC, an antigen-presenting cell. So it's presenting little bits of those antigens, those pathogenic antigens, which we drew first thing during this lecture, on the outside of its body. And this is what we call a self-non-self complex. The self part of it is, actually, we'll be discussing it in the next lecture, but it's a major histocompatibility complex, an MHC. And it's going to be attaching a little bit of that pathogen on the outside, and that's the non-self part. So a self-non-self complex. Now it turns out that T lymphocytes are specifically activated in the presence of a self-non-self complex. T lymphocytes are always monitoring the body and seeing what's going on with the cells. So the T lymphocytes will be attracted to these self-non-self complexes. And additionally, guess what? There can also be a self-non-self complex that could be produced by an actual infected cell. You'll see, technically, lung cells are not really part of the immune system, so we still draw this little self part in black. It's not really part of the second line of defense, but it is presenting that little bit of the structure that had infected it in the first place. So the T lymphocytes are attracted to this self-non-self complex also. Now what happens next is a rather complicated story, so I'll be telling it briefly here, and then I'll be telling it in more detail in the next lecture. But the basic idea is that if an infected cell presents bits of an infection on the outside, and the T lymphocytes are reading that, they're saying, okay, this is an infected cell. I understand that this self-non-self complex is basically a cry for help. The T lymphocyte is able to go ahead and destroy that infected cell, which, as you can imagine, destroying your own tissue, that's a really kind of a dangerous role, and we have to make sure that we get it right. So the T lymphocytes sometimes don't get it right, and that's oftentimes how you get things like autoimmune disorders going. But also, if an antigen-presenting cell is presenting a self-non-self complex, it's saying, okay, we have this infection going on, but remember that the antigen-presenting cell, that phagocyte, is not actually infected, it's not actually sick, and therefore it's not a plea for help, it's rather just sort of a flag waving around saying, okay, we should maybe think about mounting a further response to this particular pathogen. I think if the T lymphocytes are able to see or read enough of these flags, then there will be more actions taken. But in this case, the T lymphocyte would not actually be destroying the antigen-presenting cell. It would simply be energized into cloning itself and to keep the fight going. So these two different stories here, we're going to be elaborating on them and going into more detail in the next video, but I did want to put you in the place of sort of understanding that and giving you the idea of the self-non-self complex here and go back over it, make sure you're really understanding it, because it's only going to get more complicated the next time, so make sure you're doing your work. Right, so then we, to really elaborate on that third line of defense, remember we said it was internal and specific. The three characteristics of the third line of defense would be specific, systemic, and memory. So again, we're just bridging this gap in the knowledge here, but we're going to be going into more detail on exactly how that works in the next video. Think vaccines, though. That might be helpful to think about vaccines and how they work. Okay, let's go ahead and draw a little slice of this pathogen that we drew up at the top there. I want to draw some of those pathogenic antigens. We'll draw 4 of them. Some of them will have a rounded structure, and some of them will have a smooth structure. So we'll draw 4 of them, 2 round and 2 pointy. And it's a little simplistic to say roundy and pointy, but at the same time, it's kind of exactly how it works. This works by shapes. So here we're drawing an antibody. An antibody is a free-floating protein that is attracted to antigens. And it basically, the antibody is a Y-shaped protein, and you can see that both arms of the Y will have the shape that is complementary to a particular pathogenic antigen. Antibodies are found in the blood, interstitial fluid, and in the lymph, and therefore it's called humoral immunity because it penetrates the body's humors, fluids, but it can't actually penetrate into cells. So it's helpful to remember that T lymphocytes are able to penetrate cells and see what's going on inside of the cells. But the B lymphocyte story, which is what we're telling here, is only outside of cells, only in the fluids of the bodies. So it turns out antibodies are produced by B lymphocytes, which are most commonly found in the germinal centers, but will also be found in other parts of the body. But remember those germinal centers, we talked about those being found in the lymphoid follicles. So basically any place where you have lymphatic tissue would be a good place to meet up with B lymphocytes. So whenever you have a pathogen that comes along and you want to make sure that there's an antibody response against it, it's a good idea to pop it into that lymphatic system, make it go through all those lymphatic capillaries and vessels, and eventually go to a lymph node where it will encounter lymphoid follicles, where it will encounter germinal centers, and then the B lymphocytes can produce antibodies to go throughout the lymph, throughout the blood, throughout the interstitial fluid, throughout the humors of the body, and mount a response against that pathogen wherever it may be found. To recap the general idea of the third line of defense, T lymphocytes generate cell-mediated immunity, and B lymphocytes generate humoral immunity.